Hello students, this is Faizan Mirza. We are studying the concept of gene probes to understand how it is actually used in a microarray. So gene probes are used in microarray, and they are not just used in microarray. They are used in various other techniques such as blottings, the northern southern blottings, which probably we don't even discuss at A level. So yes, gene probes are end restriction, fragment length polymorphisms. These are various techniques in which in gene technology probably you will be discussing these later, or at least not in A level. So yeah, gene probes. These are single-stranded, short strand sequences. So for example, here I have mentioned a sequence double G double A T A C T. So this is one gene probe, and I have made two copies of it. I have attached them in two dimensions. This is along the x-axis, this is along the y-axis. So then I have another gene probe like double G, double A, double T, A, C, T, and then the same copy of this. This is a single-stranded structure. This is a single strand. This is also same copy of it attached like along the vertical axis. Then this is another other gene probe, and there's another set of gene probes. So these are all gene probes here. Now, what do these gene probes do? I can have genes, and I can just get copies of genes from two sources of organisms, and I will just have I'll just make sure that my genes that I have collected, I'll denature them. I'll just heat them enough so that the DNA molecules split into single strands, and the complementary single strand will come and attach to this specific site. For example, if I have a gene having a complementary sequence to this, it will attach here. If my gene is having a complementary structure to this, it would attach here. If it has a complementary structure to this, it would attach here, and same here. So what I do, uh, DNA fragments can be collected, or if DNA is collected, then the DNA fragments can be made. Probably you can just recall how the DNA fragments can be turned into single-stranded DNA molecules. So, for example, my DNA, I collected it from two cells. The two cells are two sources, actually two sources. So, two sources in this case could be two species. For example, I'm trying to see how close two species are related. So my DNA samples are taken from two species. I'll just make I'll make the DNA single-stranded molecules. I'll label both the DNA with a color tag, and then add them to microarray. What's microarray? A bit more of that sometime later within this video. So here, I'm tagging one DNA source that's taken from one species with a rat tag. I'm adding the DNA source taken from the DNA molecules taken from the other source with the green tag. So this is my one species DNA. This is the other species, and these are some more DNA genes from the same two species. Again, you can see I'm adding red. All these red tags, they are showing that they all belong to one species. The green actually tells you that they belong to the other species. Now, if I have these single-stranded tagged molecules, I can't see these as yet. What I've done, I will just add them to these gene probes. So when I run them to these gene probes, the genes which have the complementary, for example, the sequence is C, C, T, T, A, G, G, which is just mentioned here. So it will look for its complementary gene probe, or probably you can say the gene probe will look for it. So it will come and attach. So this is called as hybridization. So I will allow my DNA fragments to hybridize with the gene probe, and I can see that within no time I'll see that these my gene probes. I'll just run this in a bit speed. So you can see that complementary base pairing occurred, and my red DNA gene of complementary gene probe attached here, and this green is attached here, this red attached here. This is all complementary base pairing because the gene found its probe, or you can say the probe found its gene. So what I'll do, I'll just wash off the or you can say unhybridized fragments of the DNA so that they get removed from here. Now, what you can see here, you can see that this gene probe found its complementary gene in one of the source but not the other. So the green one didn't attach here. This gene probe could not find any, so this gene is probably missing. I know this gene is completely missing in this organism, in both the organisms. So yeah, here you can see the green one attached, so you can see that the red one didn't attach. So okay, so this gene is probably present only is actually present in one species only and not the other. And this gene, the gene probe here found one of the red tag and one of the green tag complementary, so both of them attached. So this means that the gene sequence present in one source of the organism and the other species, this is exactly the same. So these two organisms, they share this gene, And what's the gene sequence? I know the gene sequence because I've used a gene probe. So once this is done, I interpret this data, and the colors can be interpreted to understand what I have actually found. So this is microarrays. In microarray is basically a tool, and this tool is called as the gene chip. In gene chip, the gene's expression or the analysis of whether two species have the same genes or not, or how similar they are, we use microarray. It's a very quick and precise tool to get this information. It identifies the expressed gene in a particular cell as well. So this is a gene chip, or you can say this is a microarray. This small window here is a 2 centimeter square window. This is the microarray. This will have these single stranded gene probes attached or adhered to its surface in extremely high quantity here. So this is all molecular, so you have to visualize this really good. So this is a 2 centimeter piece of glass, 2 centimeter squared piece of glass or plastic, and short lengths of gene probes, which actually are single stranded DNA fragments. They are all adhered on the surface, and you can see that you can expect that their bases will be exposed, and they are attached in 2 dimensions, like I showed showed to you that they are attached along x-axis and y-axis, and these are the gene probes attached here. So how many? As good as there are like 1,000 positions of gene probes. These are attached per centimeter squared area of this microchip, or you can say microarray, not the microchip, the gene chip. So each portion is having multiple copies of the same. I just showed to you 2 copies of same gene probe in the previous slide. Actually, it's not 2. There are like multiple of these, extensive number of same gene probes at the same position. So they are present there. What do? You add your DNA here, and you just see whether the two sources are having the same DNA, or what's the difference there. So the advantage is that the large number of genes can be studied in a short span of time. The result is very quick. It gives an analysis it increases the amount of information that has been made available regarding whether the genes are present in an organism, or how similar they are, or just genetics of the cell. So its uses are enormous. So here you can see in this diagram, the flow chart, I've shown that the DNA is collected, and you can see that the DNA is collected so from the different sources, from the 2 different species. The collected DNA from each species is fragmented and denatured, fragmented by restriction enzyme, denatured by, you can just know, by heating and at 95 degrees Celsius. Denaturing means separating it into 2 strands. You add both different colors, the green tag and the red tag to different of the two different species. Like tag one as red, tag the other as green. Allow hybridization to occur, so you add them to the microarray. The fragment hybridize. You just place it in the laser scanner. And before you do that, you wash off the unhybridized DNA from here. So now, once you place in the laser scanner, you will see 4 different kind of spots: a green spot, red spot, yellow spot, and blue spot. You interpret this data. So how's the data interpreted? The data can be interpreted in this way. This is how the laser image of the this microarray would look like. You can see the four kinds of spots: red, yellow, green, and blue. The red spots, all these red spots, what do they represent? They represent the gene present in one species and not the other. The green spots represent that, okay, this gene is present in that species and not the other, and not the red one. And the yellow spots actually tell you that this is a common gene. So this gene is present in both the species, which were tagged green and red. So they both found their gene probes here, and they are attached here. The ones which are left blue, these are the ones which are vacant. So the gene is absent altogether in both their species. So this is how the 4 different colors in the microarray can be interpreted: the green, the red, the yellow, the blue. And again, if you just look closely here, so you can see there are spots, for example, which are of a different shade. So here, if you just look closely, you can find that there is a spot which is like yellow in color, or a bit lime. It's not exactly yellow, it's more lime. For example, this one looks orange. So you have an orange spot. Okay. And then if you look closely, for example, you can see probably just this is spot. If you look closely, this is a lime colored spot. And these are again orange spots. So you can see orange spots here and orange spots here. I have not listed orange color here. So where did the orange color come from? Think about it. So another use of the same technique is to identify the expression of the gene in cancerous cells, or cells who have a predisposition towards becoming cancerous. So you know that the gene activity or the gene expression in the cancerous cell is slightly different. That's why they become cancerous. So there is an overactivity of the genes or underactivity of certain genes. So if there is an overactivity of a certain gene in that cancerous cell, so a lot of mRNA will be produced as compared to the normal cell of the same tissue. And if there is an underactivity of the same gene in that cancerous cell, so there will be a lot lesser number of mRNA being produced in that cell which is becoming cancerous as compared to the cell which is normal. So what are we doing here? We collect mRNA from the non-cancerous cell and the cancerous cells of the same tissue, and then, and if not the same tissue, then probably the same organism, but yes, somehow of the same tissue. So what do you do? This mRNA is collected, the mRNA is converted to cDNA. You can probably recall how cDNA is made. cDNA is a single-stranded DNA molecule which is made by using reverse transcriptase enzyme, and you just add DNA nucleotides and mRNA is added, and you add the reverse transcriptase enzyme. It reverses the process of transcription, which means that rather than making mRNA from DNA, you ended up making DNA from mRNA. But this DNA is a single-stranded DNA, so it's called cDNA. Now you go and amplify this DNA using PCR technique, and once the DNA has been amplified, you just fragment both these. You actually fragment them using restriction enzymes, and then you add color to it. You add green color. You tag the non-cancerous cells, the mRNAs or the cDNAs taken from the non-cancerous cells as green, and the cancer cell cDNAs, which are actually the mRNAs of cancer cell, they are tagged as red. Now add them to microarray. Allow the fragments to hybridize, wash off the unhybridized fragments, place this in the laser scanner. Again, you will see four different colors, four clear distinct color: green, red, yellow, and blue. And you can just interpret red means that the gene is present in the cancerous cell and not the non-cancerous cell. Green spot tells you that it's in the normal cell and not the cancerous cell. Yellow spot tells you that this gene is present in both the cancerous and non-cancerous cell that's expressed there, and blue spot means that it's not being represented in either cell. And the color intensity, the intensity of the color, it shows to you the expression of the gene, how much the gene has been expressed. So this is the flow chart, shows you cancer cell, mRNA, RNA taken out, labeled red, and hybridized with microarray. Normal cells, extension of RNA, labeled using green tags, and then added to microarray. So this is the kind of image that you can get. Again, you can just, if you look closely, these are some orange fragments and orange spots, and you can see this not just the orange spot and this yellow spot, lime spot as well. So why are we having different colored spots? So because of the intensity. So if this spot here is an orange spot, this orange spot tells you that on this gene probe, more cDNA, which was tagged red, found its way here, then green. So if you add a lot of red color and a little of green color, you get orange tint. So the orange tint actually represent that for this gene, this has been overexpressed in the cell which is cancerous, and not expressed in that abundance in the normal cell. And for example, if the spot I'm talking about is lime, it's more towards green side. It actually shows to you that the RNA or the cDNA taken from the cancer cell is a lot less in number as compared to the one taken from the normal cell. So this brings us to the diagnosis of predisposition towards cancer. So females who are, or even males, but we will be restricting our discussions to females mainly. So breast cancer. Predisposition towards breast cancer can be screened by screening a female for the overexpression of two genes, BRCA1, BRCA2. So BRCA1, BRCA2, if either one of them have been overexpressed, and you get an orange spot in that case, so that means that the female is predisposed to familial cancer, primarily the breast cancer and the reproductive cancer, the cancers of the reproductive organ, the uterus and the ovary as well. So if the screening shows this to you, so the female is then asked to go for elective mastectomy, and this is just to make sure that she won't develop the cancers of these tissues in future, because she's already predisposed, because these genes have undergone overexpression, and overexpression means that it's just a matter of time that this kind of breast cancer would actually develop. So to prevent the development of this cancer, because it's just impending there, waiting to just show itself, probably in some years. So the female is just asked again. I'm just explaining this to you, that she's asked to go for elective mastectomy, and this is the surgical removal of the breast tissue. So if the breast tissue is not there, then even if the BRCA1 and BRCA2 genes have overexpressed, since the tissue is not there anymore, so it won't cause cancer. So this slide here actually sums up all these 7 points here. Actually, they tell to you what each color is actually representing on a microarray, and how this can be actually turned into, and how the data can be interpreted, and what information can you gather, and how you can correlate this information and lead to some fruitful diagnosis. That's it from my side. Thank you so much.